Okay, so our next speaker is Ana Raquel Díaz. Ana Raquel holds a PhD in organic chemistry from the University of La Laguna. Her scientific career has been focused on the research of substance produced by organisms that inhabit marine ecosystem, algae, invertebrate and microbes organisms, the isolation and determination of their chemical structure, their ecological role, and their possibly biomedical application. After a postdoctoral period of 3 years at the University of British Columbia, Canada, she continued her research in the area of marine natural product, IPNA-CSIC, and at the Instituto Universitario de Bioorgánica Antonio González, University of La Laguna. Since November 2021, she is a tenure scientist, a member of the Chemistry of Marine Natural Product Group at IPNA-CSIC. So you've got 20 minutes, and I'm going to put your presentation.
Thank you, Eugenio, for your presentation. And I will also thank you for the chance and for the invitation to be here and the chance to share with all of you the research I've been doing around the volcanic habitats we have at the Canary Islands. So as you mentioned in my presentation, I just been for 1 year permanently joined to IPNA, but all this work I'm gonna explain you today has been or started at the University of La Laguna. So for you to understand a little bit our interest and our approach, our group is a marine natural products group. So we are interested mainly in the molecules produced for organisms coming from marine sources, both macroorganisms and also microorganisms. Our role in the laboratory is isolating these compounds as well as looking for, elucidating their structure and evaluate their pharmacological potential to look for other alternatives for those compounds. Aside from that, we also are interested in culture and marine biotoxins produced by dinoflagellated microalgae. But an important point in all this research is also habitat, and that is what I'm going to talk about today, because of my interest in exploring the microbial biodiversity of volcanic habitats. All of you probably know that marine ecosystems are highly biodiverse, and the organisms in habitat in these environments need to compete, especially benthic organisms which are attached to the floor or have very limited movement, need to compete for food, for space, and their relationships between organisms from different species and also from the same species come from, they require a chemical mechanism for that relationship. And the way they do is by producing compounds called secondary metabolites, and that are also known as natural products. So that's my areas of expertise, and those are the compounds we are looking for. These are some examples for you to have an idea of the sort of molecules I'm talking about. They are not big molecules. They are species or genus dependent. And as you can see, they have the singularities, like the presence of rings and functional groups and presence of chains. allows them to have a three-dimensional structure, like that one represented here by Staurosporin, which was isolated from a mangrove, Streptomyces. So this three-dimensional structure these compounds have allows them to interact with biological targets or enzymes that confers them allows them to have biological properties. That means that these compounds are produced for those organisms to have an ecological role, but at the same time we can take advantage of that role to look for additional biological activities. So that's what we are looking into microbial macrobiota coming from the oceans, and especially those coming from extreme environments. So that's because of the capacity of microorganisms to have to, because of they are sensitive to the extreme conditions of the ocean, and they react to those conditions by biosynthesizing compounds and unique metabolites. So extreme environments are considered polar environments, but also deep sea ocean and hydrothermal vents. And that's something that we do have in our Canary Islands. And especially in the last two events occurred in the island of El Hierro in 2011, and also last year in La Palma. So that's a privilege, but for a laboratory of natural products of chemists, that means a problem, and you need because you need to access to the infrastructure, and that is the opportunity that Eugenio gave me in 2016, 5 years later, after the volcanic eruption occurred in the island of El Hierro. So I participated in this oceanographic cruise in 2016, and out of that we were able to sample the different organisms and these different environmental samples out of 7 rock dredges that were taken as you can observe here in this bathymetry. So in the end, we collected a total of 30 samples from depth ranging from 160 and 800 meters depth. At the same time, the physical chemical data were collected using, as Eugenio showed this morning, a rosette, and where the anomalies, pH anomalies and temperature anomalies are detected close to the main crater and a secondary crater, which are these samples here recorded that you can see in this table. Here in this column, you can observe how heterogeneous were the samples that we collected, and you can see it also in these two in these photographs, A and B. So attending to that nature, the samples were surface sterilized and then processed, attending to their characteristics. So for these sort of samples that are microorganisms, we try to isolate microorganisms associated to the inner tissues of those of those samples. So once processed, the samples were transferred into Petri dishes, as you can see here. We used 2 different media. We decided to use an enriched media and more oligotrophic media. Maybe next time I'll try to add some of your ingredients to see if we can get some other bacteria. Well, the Petri dishes were incubated at 25 degrees Celsius and were monitored for a period of six months. During that process, all bacterial strains were transferred to new agar plates. As you can see here, they are the colonies, but they are mixed. They are in a community. So you for the isolation, you need to transfer them to new Petri dishes. And in the end, what we got was a collection of isolates, bacterial isolates, that were identified using the methodology that Jesus just described a while ago. The only thing is that we isolated the genomic DNA for each of these isolates. We use again the 16S ribosomic RNA gene using universal primers, and the sequences obtained were compared with those showed in the databases. So we obtain for our collection this map, where you can see in the outer ring the genus identified and the classes these bacteria belong, and the proportion of all of them. As you can see, the representation of protobacteria is quite high and is coherent with data that Ferreira and Jesus just mentioned. From the 84 sequences that we analyze, we perform a phylogenetic analysis, as you can see in this tree, with that in general terms are distributed in 28 genera that belong to 4 phyla. With, as I mentioned, the main representative are proteobacteria. This purple color represents the gamma proteobacteria, and Firmicutes is blue, and the actinobacteria are in red, green. One aspect is that while most of the isolates we analyzed had a high homology with higher than 90% with those found in the database, we have a family of Vibrio which homology is lower than 97%, which are indicated here with arrows. So once we have the collection of bacteria, as you remember from the beginning, our interest is looking at the chemistry they produce and also looking at their biological activity. So what we did was selecting around 30 bacterial isolates belonging to 20 genera, and we transferred them into liquid culture. But what we did was culture each bacteria in 4 different conditions. That is called OSMAC, a strategy that comes from the acronym, is the acronym of one strain, many compounds. What we did was using 2 different culture media, and we changed aeration conditions and nutrient concentration. In all of them, we kept constant these two parameters, temperature of incubation, and we cultured them for a period of 30 days. Once this period was over, we used organic solvents to make extraction of the chemical production that these bacteria produce, and analyze the antiproliferative activity. For that, we have a collaboration with Dr. Padron that comes from the biolab at the University of La Laguna, and he and his team tested the activity of the extract in 6 different cancer cell lines. Here you can see an extract of those results, where here in the graphs you can see this color. Each color represents one of the cell lines, or each cell line, and this is the growing inhibition of half of the population of the cells. As you can see, the average values we obtained was 50 micrograms per milliliter, which is quite an acceptable activity, but we focus in the most active values that are here represented in this table. And as you can see here, we have some examples where the biological activity goes below 1 micrograms per milliliter, which is quite promising. So that at this point, we have the selected which are the promising strains, but also what are the culture conditions to get the activity. If we analyze the effect of the culture conditions and relate it with the antiprolifer activity, that's what we can see in these radar charts, which represent the minimum growing inhibition, and here the maximum volume growing inhibition of every strain isolated and tested in the 4 different culture conditions we tested. The conclusion at looking at this chart is that the least nutrients we use, the best results we obtain for activity. This is more or less the same conclusion, but looking at each of the cell line independently. So at this point, the next step, which is currently ongoing, is with those selected and promising strains, scale up the cultures to have a look into the chemistry they are producing. So these photographs show a large-scale culture. This is around a 10 liters culture. This is after 7 days, but we let the cultures go for 30 days, and you can see how the appearance changes after time. Once the culture finished, what we separate in this case the cells from the culture media and prepare an extract from both the biomass and the culture media. At this point, we haven't continued with the chromatography process to analyze the chemistry, but we have confirmed that the scale-up kept the biological activity. Here we have it, and it's found in the biomass extract. However, in the culture media, we don't get any activity. And moving to the next chapter. This is I'll be quick. we had just the chance to jump for a couple of days also in February this year to take samples from the area affected by the eruption in La Palma. In that occasion, we use a different strategy for sampling, which we use the ROV, and we sample at the lava delta and also in front of the coast of La Bombilla. These are the depths. They are less deeper, organisms less deeper than the previous work. And this is a summary of what we've doing. This is the biodiversity we found during sampling, which was to me quite surprising. And from these samples, we have started to process and the isolation process. And for now, from the 29 samples we got, including the ashes and sediments deposit on the bottom, rocks and invertebrates. We have already a collection of 80 bacterial strains that were isolated from these two different media. And that's it. Just would like to finish with a thought, and it's the importance of the exploring the microbial biodiversity of extreme habitats, as the volcanic submarine habitats we have in the Canary Islands. They represent an excellent starting point for development of biotechnological tools to access chemical compounds and substances that may have potential industrial application. And also another thought is the importance of institutional support and collaborative and multidisciplinary perspective to make this research possible.
Thank you so much, Ana Raquel Díaz from IPNA-CSIC. Actually, I wanted to say that the ocean is a lot of substance over there that is just waiting for us to discover. So thank you for your work, because it's quite important in order to increase our society, our healthy and everything. So it's a really good job. Thank you so much for that. Any question in the auditorium for Ana Raquel?
You said that you isolated, like, from the samples 182 bacteria, but only 84 were identified. And I would like to know why. Maybe there are some that cannot be identified, or that you chose some for a special reason, or they were like just repeated samples. So
yeah, that's a good question. I was too quick to mention it, but yeah, it's true. We should have analyzed all of them, but what we did was selecting or just not considering those that came from the same original sample, those that have the same appearance, just to avoid replicas. That's the reason why.
Any other question? The auditorium? Online?
Yeah. I mean, first I must say, wow, what amazing 2 data sets. Your team must have a lot more patience than I would in that sequencing. I guess this is kind of a classic question, but what's next? Are you going to look at other volcanoes? What's next?
Well, just with these 2 collections we have right now, we have a lot of work in front going on. This is just the beginning, because as I mentioned at the beginning, our interest is looking at the chemistry these organisms produce. So now what we are doing is scaling up, as I show in the example, make big cultures and then start analyzing the compounds that they do. In the first part in El Hierro, we tested the activity against cancer cell lines, but my idea is also testing other biological activities. For example, we have collaborations with a group that looks for anti-parasitic activity. And in the end, what we are looking at is giving a potential, an opportunity to look for something that can be a benefit for society.